Wanted to thank the organizers for the chance to talk about some of the work we're doing in the lab. It's been already a really interesting workshop, so I'm learning a lot. So I wanted to start off with a metaphor. So when planting a seed, it's not just important to think about the seed itself, but also the soil and the nutrients that are available. So in order to actually see a plant grow, we need to consider the nutrients within that environment. And we're also thinking about this same concept for the gut microbiome, because we're really interested in engineering this ecosystem and having predictive control. And so when we think about introducing engineered probiotics to change functions of the gut, to influence various aspects of human health and disease, we also need to think about the diet and the nutrient environment. So we need to kind of engineer the environment at the same time that we're engineering the ecosystem. And so we can think about these dietary factors or nutrients or environment in general as a control knob, just as we think about engineering the ecosystem as well, the bacteria that are present, introducing new microbes in. So we shouldn't expect probiotics to establish or persist unless they have the right niches available. And so the way we've thought about this system, this really complex system, is thinking about synthetic communities. We build communities in the lab. We use high throughput techniques where we can array combinations of bacteria and environmental factors. And this helps us explore really large design landscapes. In the case of just 25 species, we have about 33 million possible combinations of species that we can study in the lab. And then for 100 species, it represents roughly the number of species in our gut. We have about 10 to the 30 possibilities, which is impossible to exhaustively enumerate, even with the most advanced high throughput techniques. And so microbial interactions are really complex, and they have many different mechanisms by which microbes interact. We think a lot about the metabolites that are mediating these interactions and mediating interactions with the host. And so conceptually, we can think about this complex mapping between the genetics of the community, weighted by the abundance. This could also be measurements of function like transcriptomics or proteomics, and the extent to which this community performs a function. And this could be a very rugged landscape, could have many peaks, valleys, and we're often trying to navigate to peaks in the landscape so that we can optimize the functions of these synthetic communities as probiotics. And we want to identify how to navigate this really large landscape given the sheer complexity of the number of combinations that we have in the system. So another challenge with studying these microbial communities is that they are highly nonlinear. They are very dynamic. They're constantly changing in response to environmental stimuli. This makes prediction and design very challenging. Also, our measurements are very noisy and limited. So we can't really sample the system very often, especially when it comes to natural environments and natural samples. And so this necessitates uncertainty quantification, where we can use computational models to predict the behavior of the system, but we also quantify our certainty in those predictions. And so we use Bayesian inference to capture that uncertainty, and we use that for experimental design techniques. And just to illustrate what I mean, so if we have on the x-axis possible experiments that we could do, sorry, I'm trying to figure out this. For some reason, it's showing up, but okay, yeah. So on the x-axis, there's different experiments that we can choose from. And some of those experiments have, we have a lot of confidence in the outcome. So the y-axis represents what we're trying to measure about the system. That's our objective value. And so in those regions where we have a very low uncertainty, those are not measurements worth taking. But the measurements in red, which have high degree of uncertainty, represented by that envelope around the line, those are measurements that we are not confident in, and so that would give us the most information if we took those measurements. So we can use this uncertainty to guide experimental design and select which combinations of environmental factor species that we could study in the lab to maximize our understanding of the system, fill with knowledge gaps in the model, and also satisfy our engineering objectives. So the short kind of vignette that I'll tell today is about using these types of approaches, active learning, to navigate large design landscapes where we're interested in optimizing not just the species as a probiotic, but also the fibers together. So simultaneously trying to identify these combinations of fibers and species that will work together, enhance colonization and the functions that we're interested in. And so using these in vitro approaches, we can take these tens of millions, hundreds of millions of different combinations that we could build in the lab and funnel it down to just a smaller subset that we can study in more realistic environments like germ-free mice, for example, that we can introduce microbes and also control the diet as well for these mice. So we can really precisely control the environment. So we've been really interested in metabolites that these microbes are producing. And for some reason, this thing is like not showing up very well. Can everyone see my laser pointer?
No.
Okay, I'm not going to use it. So we've been really interested in metabolites because they are a major variable by which the microbes interact with the host. And within this like complex community of hundreds of species, each microbe has, metabolic pathways and also has regulation. And I just wanted to give a little shout out because I'm not talking about it, but maybe Chris will give a talk tomorrow on this. But regulation is also really fascinating. I'm not going to talk about regulation today, but just in one particular example, we were able to show that regulation, if you consider the complex nonlinear regulation for a really simple community, you can get these really beautiful limit cycle oscillations over long time scales. So I'm not going to talk about regulation today, but hopefully Chris will talk about it and focus mostly on metabolism. And so the microbes are producing these metabolites and shaping the ecosystem, but also shaping interactions with the host. And so these metabolic networks are really linked and interconnected between the host and the bacteria. So when we thought about simultaneously designing fibers and bacteria together to alter and control gut health, we designed a synthetic community of about 15 bacteria that are highly prevalent across the human population. And we also selected a set of fibers, 6 chemically diverse dietary fibers that vary in terms of their chemical structure and their complexity. So how many bacteria in the gut could use them is related to complexity. So do they have these complex polysaccharide utilization loci to degrade these complex fibers? And so we did a series of experiments where we use active learning, so design, test, learn cycles, where we take experimental data, we build a model to predict the temporal change in community composition and metabolites that we're measuring. And then we use the model to predict all possible experiments and then select the experiments that balance exploration and exploitation. So exploration is filling knowledge gaps in the model by leveraging the parameter uncertainty in our predictions, and the exploitation is maximizing an objective. In our case, we were interested in 3 components: how much butyrate the community produces, which is a healthy metabolite for the gut, diversity of the community, and then what we call stability, which represents how much temporal change we see in the composition over time. And so we have a weighted objective of these 3 different components. And so we do a series of experiments. Each experiment consists of about a couple hundred conditions. And so the design space, if we consider all the different combinations of fibers and different levels of those fibers, we have about over 15 million combinations. So we can only select a really small subset to really measure in the lab. So that's where we're leveraging, objectives.
You can imagine why the stability diversity might be linked.
Yeah, that's a great question. So we have looked at the relationships between objectives. Butyrate and diversity are kind of weakly correlated, but they also have this trade-off where communities that have the highest butyrate are also not the most diverse. So there is this kind of like front to the relationship between those. I'd say a couple of them are completely not correlated, like stability and diversity, I believe. But yeah, I have to look back on that. But we have looked at the relationships between them. None of them are very highly correlated.
Say stability and diversity are not correlates?
Yeah, well, we were measuring stability is looking at how much the composition changes, essentially looking at a distance metric over time. So that's not necessarily directly related to diversity. So there's really not a strong correlation there. Yeah.
When you measure butyrate, are you measuring a production flux or a concentration?
We're measuring a concentration over a fixed time window. So you can calculate a flux or a change over per unit time, but it's, you know, likely changing dynamically over time. We are measuring it at every time point. And we're modeling that change over time. Yeah. Yeah.
Stability, I should think of it as like a time scale on which things are.
It's how much fluctuations or variability you have in composition over time. Maybe there's a better way to phrase that. It's more temporal variability in composition. So for example, if you go from having like one species dominating to like many species growing, that would be a community that's less stable versus a community that stays very constant over time. Yeah. Yeah.
So then butyrate and diversity are moving over time, but stability is your measure is it one or is it over a time window?
We can calculate it between time points. I think we primarily used the two final time points because it kind of represented where the system was approaching a steady state. It should therefore be more stable. But yeah, we can calculate it between time points. So what I'm representing here is the experimental measurements, where every single data point represents the different combinations of fibers and bacteria that we inoculated. And then all the colors, the different colors represent the design, test, learn cycles. And so as we proceed through the experiment, or experimental process, we end up converging on these green points that represent, they're more similar to each other. So that also, I should also mention, the distance between the points represents how similar the phylogenetic tree and initial inoculums are in terms of fiber and species. So points that are closer together are more similar in terms of the types of fibers and types of species. Points that are further apart are very different. So really converging on this kind of more narrow part of the landscape. And then we can show the same plot, but now looking at this objective value, which again is like a weighted sum of these three different properties of the community. And in the cluster that's more similar to each other, we have higher objective values. And so we are converging on at least local optimum of the landscape and some sort of optimum of the landscape. And so we identified this subset of communities that's pretty similar to each other in some ways, but has very high objectives. I should also mention we only explored about 0.008% of the landscape. So using this trained machine learning model, it's a recurrent neural network. It's been modified to capture more physically realistic outcomes. So it's tailored for microbiome. It's Bayesian recurrent neural network. We can perform this explainable artificial intelligence, which is just analyzing the effects of different inputs on the system's output, so the prediction. So every single measurement we have, different species and fibers, has a different SHAP value. So SHAP tells us whether that particular species or fiber is enhancing butyrate or inhibiting butyrate. And this again is a statistical analysis and should not be interpreted as like a direct effect. And so what I'm showing here is each individual species on the top plot and fibers on the bottom plot. The y-axis represents the SHAP value. So positive interaction, negative interaction. And we're specifically looking at butyrate here, but we've looked at all aspects of the objective function, all three components. Because butyrate we understand about, we know a lot about it. And so as expected, we find that our 5 butyrate producers have positive SHAP values overall. And again, we have a distribution across all the different communities fibers that we measured. We did find this robust motif where Acacia is producing a lot of butyrate and inulin and B uniformis enhance the butyrate production of Acacia. So this motif of inulin, B uniformis, and AC is robust across all the different combinations we measured, about 2,000. And then we also find some interesting interplay with Prevotella copri that also emerges from the SHAP analysis, where Prevotella is able to enhance butyrate production. But then when Prevotella, Copri, and B. uniformis are together, we see this kind of higher order interaction where Prevotella and P. and B. uniformis lead to a reduction in butyrate. So these two species alone enhance butyrate, but when they're together, they inhibit butyrate. And so we were interested in validating this, and we collaborated with Sean Gibbons to do some genome-scale metabolic modeling of the interactions between Prevotella, B. uniformis, and inulin. Unfortunately, we can't look at, B. uniformis, acacia, and inulin. We can't look at Prevotella because Prevotella is missing the inulin utilization loci, our particular strain. So we can't add that to this model. But using the genome scale metabolic model, flux balance analysis, we were able to recapitulate this positive interaction and then generate a hypothesis about how BU and AC are interacting to enhance butyrate production. We also performed an experiment where we just cultured the individual species and their pairwise combinations and the triple. And we're able to confirm that this is indeed some sort of higher interaction where Prevotella and B. uniformis are inhibiting butyrate production relative to when they are in isolation with acacia. So this is a really interesting interaction. It also seems to shift towards propionate production. And so we're interested in digging into this further. So this confirms that our SHAP analysis can identify these higher order interactions that typically when we use models like GLV, we are unable to capture. We were also interested in testing to see how our engineered communities and fiber combinations perform in the context of the mammalian gut and also in human fecal communities. So those are two separate experiments that I'll talk about now. So there's three properties we're looking for. We can't directly measure the same things as our objective function. We can get close to it, but not exactly. We were interested in colonization. So how well does our community colonize the mice in the presence of these design fibers? We're also interested in predictability. How much predictability do we have going from in vitro to in vivo? And then finally, can we alter the functions? Can we change the short chain fatty acid profiles, which are again linked to host health in a variety of different ways? And so we had three different treatments or conditions. One of them is our optimized, highest performing symbiotic, this combination of fibers and species, which we call IPX HiCom. And then we have our lowest performing community fiber combination, which we call low-com, which is with Xylan. And then we have a community that doesn't have any microbial accessible carbohydrate, which is cellulose. It's also with HiCom. So HiCom is a community, but the mice do not have the bacteria don't have a microbial accessible carbohydrate. And so what we're looking at on the first plot is a Shannon diversity. And so we can see that the HiCom IPX condition, the symbiotic, has high colonization relative to low-com. And then in the case with colonization, our model optimized symbiotic has the highest colonization of the three treatments. So that was consistent with what we were hoping for in the outcomes. We also looked at the community composition, and we saw some similar trends and some differences. So if we look at this plot over here, this is the 96 well plate. So this is the in vitro condition, and this is the in vivo condition for low-com, and then in vitro and vivo for high-com, and this is just the cellulose treatment for high-com. And so we see some similar trends between in vitro and in vivo. And if we quantify the distance between the in vitro and in vivo, both fiber conditions reduce that distance, therefore improving the predictability. So when we have fiber, the community behaves more similar to in vitro versus if we don't have a microbial accessible carbohydrate. So that's what this plot is showing.
Color taxa or species?
Sorry, each color here is a different treatment. There's different taxa. Yeah, so these are different members of the community, each color. So I don't have the legend here. So, so then what was really striking is when we look at short chain fatty acids, the trends in vitro and in vivo, again, showing the in vitro, I'm over here in vivo, vitro, in vivo, in vitro, in vivo. are very similar. Butyrate was not statistically significant. Butyrate's really hard to measure, but there's a trend that's very consistent with in vitro. But propionate and acetate have a really striking trend in terms of which community fiber or not fiber condition was high or low. So we see very similar trends between in vitro and in vivo. So we were able to use our in vitro active learning process pipeline to explore this large landscape and then optimize these communities that we can introduce into mice, and then we see very similar trends. So another experiment we were interested in, this was a reviewer question, which I thought was a great reviewer question because it pushed us into this new direction, was could we take fecal samples and invade with our symbiotics and show consistent results? So we got fecal samples from a collaborator from obese pediatric patients, and we cultured them in the lab, and then we have a series of treatments. So we have a no intervention case where we don't really add anything. We have our symbiotic, just the community alone, and the fiber. And we culture these communities over a period of time, and then we perform metabolomics, sequencing, and measure OD. And this was led by Manasi, postdoc in the lab. And we have 10 different colors represents the different fecal samples. And so, which was exciting is that our high plus IPX, which is our model optimized community, has the highest butyrate across all the donors. Low-com plus Xylan is lower significantly. And then when you just have the community or just have the fiber, you get substantially lower butyrate. And then we weren't really interested like specifically in what the outcomes of propionate and acetate, but they trend with what is expected in terms of the, so yeah, propionate and acetate had trends as expected, considering that acetate is converted into butyrate and propionate sometimes has a trade-off with butyrate. Am I out of time? Yeah. Okay. All right. I mean, I think basically I'll kind of wrap up here. This is the end of this story. The last thing I just wanted to mention quickly is related to this topic of how do we model microbial communities? How do we capture mechanism? How do we capture the unknown aspects, like all of these aspects such as microbial production, pH-based mechanisms, many mechanisms that we don't understand, right? So metabolite-based resource consumption and cross-feeding is just a sliver of what microbes are doing to interact with each other. So we've developed a physics integrated machine learning model that's currently under development. So we have our first preprint out, but we're working on a new version too that's incorporating more physics. So I really appreciated the talk this morning on like how to incorporate like redox into these models. And so I won't go into details because I'm out of time, but essentially we have a multiplicative structure where we multiply a neural network by a mechanistic component. And this allows us to have the flexibility to capture these unknown mechanisms, host microbe interactions, microbe interactions, but also gives us the interpretability that we can look at the parameters and gain direct causal insight into how these microbes are interacting and what is this like transformation matrix, consumption to production matrix. So we can actually infer that from data. And I won't go into detail about it, but I'm more than happy to chat with folks about the model. We're also thinking about how to make integrate more physics into this model. And essentially what it does is allows us to capture complex patterns in the data while also allowing us to infer what these interaction networks are, micro metabolite and micro metabolite, right? Because metabolites can be converted into each other. So we've applied this model to both in silico and experimental data and validated its performance, both for interpretation and for prediction performance. And so I'm just going to wrap up here because I'm out of time. Just as a conclusion, this active learning approach allows us to navigate these very large design landscapes so that we can identify what are the most interesting combinations to study. And then we can use that to translate into more complex environments. And combining mechanistic and machine learning models is a very powerful approach that allows us to balance both flexibility and also interpretability. And I'd like to thank my group, especially Manasi Ely, Bryce, and Sharon. And I'm happy to take any questions if there's time.
There is time for 2 questions. If there is a junior person to go first. Yes, please.
Very nice talk. The question is like, your machine learning approach actually tells how exactly community works in the presence of fiber. What happens to this community when you put it on top of fecal sample? Whether these bacteria persist, like I can see the metabolites actually changing, but whether these bacteria actually persist or something more dynamic happens.
Yeah, so this has been a journey into analyzing natural samples with 16S, which is something that our lab has not done, surprisingly. So we have the data finally with this, and it does look like our community members are kind of persisting at the endpoint, which is interesting. So we actually see our synthetic community members in the fecal samples, but I don't have a complete conclusions from the data yet because we just finished analyzing it. But yeah, the conclusion is that we do see persistence in some cases, and it'll be interesting to see if the persistence is correlated with the metabolites. So we don't know. I don't know that answer yet. Yeah.
All right, Casey.
Yeah, I mean, it's really fascinating to think about the implications. Maybe I can ask you to speculate a little bit. So the picture I have in my brain is sort of like guilds that you might be discovering, but then it's sort of an analogy to, in neuroscience where you try to break down, subnetworks, and oftentimes what you find is that, there are many possible solutions to the same objective function. So I guess I'm wondering, both, do you think that you are finding the solution as opposed to like, a sort of very large landscape of solutions? And do you think that these solutions, kind of the way that your fecal sample experiments are starting to get at, are they in some ways additive, such that now if you optimize for a different objective and then you start to try to combine them together to achieve multiple objectives at once, are they going to clash, or are they going to, in some sense be independent?
I think I understood the first part of the question. Maybe a second one I might ask clarification. But so the first part is, are there like multiple peaks in the landscape? And we did some clustering analysis of our data, and we kind of showed that the communities, the highest performing communities, have very similar behaviors. We did find, very similar compositions and fibers. We did find like maybe one example of another cluster that might have high objective, not as high as the highest, but different patterns. We are now using techniques called diverse optimization so that we can look for these different peaks in the landscape. Because I think that's really interesting. Like what works for one person might not work for another. So if we can find all the peaks in the landscape, then we can have multiple solutions to the same kind of objective that we're looking for. I'm not sure if I fully understood the second.
Second was just, let's say you apply this approach for objective 1, and you do the same thing for objective 2. And so now you have sort of, in some sense, like a community composition that potentially could solve both problems. Is it really the case that they will sort of be able to act independently in the context of the whole ecosystem?
Like you have different like interventions, let's say, that you're introducing one for one metabolite, another for another metabolite, and would they kind of collide? I mean, from a kind of ecology perspective, there's limited resources. And so if they were each provided their own orthogonal niche, then maybe they could persist and perform their functions. But also there's all these correlations due to metabolism too, like fundamental trade-offs with how much metabolites can get produced. And so, yeah, that's a really interesting question. Yeah. Mm-hmm.
If our next speaker gets set up, we're going to take one more question. Last question.
One quick question. We might not have the answer. So I noticed that high com had a high persistence and low com didn't really have much persistence. Like, do you know any reason why that might be? Because I'm thinking, what would the objective function have to do with the persistence inside the rat gut? But I have.
Could you like what you mean by persistence?
When you said persistence, that means like how long it stays in the gut, right?
Yeah. So we've only done invasions with fecal communities in vitro. So we can examine persistence in that case, just how long our synthetic community members are around. In vivo, we are working with a synthetic community. So most of the time, most species engraft into the gut, and sometimes we'll have some species that don't. But there's enough like niches available in the mammalian gut that most of the time we see a pretty diverse set of microbes that colonize.
Misinterpreted chart,
Yeah.
sorry.
Yeah.
Right, let's thank Ophelia again.